It's a really cool process, and one of the most, I think it's one of the most interesting bits of coccolith biology. And people who aren't coccolith experts, is something which is worth learning about. It's a very interesting biomineralization process, but it also, as well as being a really interesting process, it's an important one for anyone who wants to understand and work with coccoliths, because it is critical to understanding their morphology, how they appear, and also it's a key to understand their taxonomy. So if you're working on coccoliths, it's really an aspect which is worth learning as well. In this talk, obviously it's going to be a fairly swift run through the main aspects. There's quite a lot of, there are some references as well, which will be in the talk online, on the version you can see online, and in the PowerPoint online. And it's, if you're working on it, it's worth going in and reading some of those references. But basically, though, we'll go through, let's say, the overall structure. So first of all, the VR model, which is the basic form of classical coccolith structure. Then some of the complications of that, and finally go into holococcoliths and life cycles. So to start at the beginning, it should actually be with Coccolithus pelagicus, not Emiliani Huxley in this case. It's Coccolithus pelagicus. It's a common species in the North Atlantic, up in temperate and colder latitudes, and it's a relatively large coccolithophore, and it is the original. It was the very first species to be described. It's where, and this is why it's called Coccolithus, and it's the type coccolith. It also, interestingly enough, was the very first species on which anyone ever studied how coccoliths form. This guy Dixon in 1900. Looking in light microscope at Coccolithus pelagicus cells, then saw, you could see inside the cell coccoliths forming, and he deduced then the coccoliths formed inside the cell, and then were shoved out to form the composite coccosphere, which is indeed what happens. On these images here, we have, here's a typical picture. And actually, it's not that easy to see them inside. You have to really focus through carefully. But one thing you can do is decalcify the cells. You just bubble carbon dioxide through a culture medium, then the coccoliths are dissolved. And actually, it shows one of the curious side effects that basically coccoliths, although they're a really important part of coccoliths in terms of morphology, and the cell is highly adapted to form them, they aren't actually essential. They don't form a physiological function. So it was a reflection of why, one of the reasons why one infers that they probably are actually predominantly protective in function. Anyway, you can decalcify them, and then you can see the cells inside the cell. It's a remarkable piece of work by Alison Taylor and colleagues, which is in a paper 2007, and they have this beautiful video, which I will show you now, which is, again, it's on the INA website, and it's actually, again, it's really easy to find on the web if you want to. If you just search for coccolith formation video, and you'll find it easily on YouTube. Or you can run this video now, which is basically, it's an 8-hour, it's a time lapse of looking at Coccolithus pelagicus cells over an 8-hour time period, where the cells were initially decalcified. So here's these decalcified cells, which then it just formed one coccolith, and now you can see another coccolith forming inside the cell. It's getting bigger. It almost fills the entire cell, in fact, at this stage. Must be uncomfortable. And then it gets pushed out to the outside, and another coccolith starts forming. You can see how it starts off as much smaller, and again it grows, and eventually it will be pushed out. Exocytose is a technical term for that. And here we see a fourth one just starting to form. That's it. So it's making about one coccolith every 2 hours in that sequence. We will progress on. And remarkable process, even just look at it like that. You see how they form, so coccoliths forming inside the cell, then being pushed out. It's quite an unusual way of doing the biomineralization. It's quite cool in terms of the final skeletal morphology, because it produces this cell which is really strong when it's crushed, when it's pushed together, but on the other hand it can grow and get bigger, unlike quite a lot of types of exoskeleton where it essentially is a rigid exoskeleton. In order for the organism to get any larger, it needs to make a complete new shell. For the coccoliths, it's quite a cool structure because basically they can add extra components at any time and make the cell bigger and bigger without having to throw away the old ones. And indeed, when the cell divides, then the daughter cells each keep part of the coccoliths, and so they actually recycle them. And the coccolith, in principle, could actually be much older than the cell. However, the cool part, in terms of how those coccoliths are forming, it's a two-phase process. First, they nucleate. There's the initial phase of calcite nucleation, which is just a very simple little rhombohedral crystals form. You can see them here. This is in Emiliania huxleyi as a sketch section, and then these are transmission electron micrographs, thin sections, showing it forming, then getting larger. And that was one way in which they've been studied, by looking at thin sections like this across cells. You can also, if you take a culture of cells, then you will end up with having coccoliths at different stages of formation, intracellular coccoliths at different stages of growth, and you can basically then just compare them, which is what we did here, from a paper in '91, where we basically got Emiliania huxleyi again, and then here are the coccoliths at different stages of growth, and you can see them going again from very simple initial nucleation of crystals, call this a protococcolith ring, then the crystals growing in different directions to form the final form. And you can see it again shown in these diagrams, which are based on a combination of these sorts of observations and SEM observations, trying that. So it's one of the advantages of getting a growth sequence, so you can really start working out how the structure works. And you can see very rapid that it started as very simple crystals, but then as they grow, they start kinking and overlapping with each other, and so the structure actually locks together. And one consequence of that is not only does the nucleation phase determine exactly the number of crystals in the final coccolith, there's no subsequent nucleation, but also it determines the crystallographic orientation, because they don't rotate or change orientation afterwards. Basically, they're nucleated in one orientation, and that orientation will stay as they grow and get larger. And you can see that in this case they're growing in. So you've got the initial crystals forming, and they grow inwards to form the grill. Oops, going back. Upwards, and then over to form the distal shield, and outwards to form the proximal shield. And they're also then, in the case of Emiliania huxleyi, they're actually growing a second. So they're forming the tube around the central area grows in two different directions. One growing that way, and one growing up inside, growing in this direction, which actually helps lock the whole structure together, which is quite typical. When you look at the coccolith structures, it's often there's quite a lot of modification of the form in order to make a rigid structure. And then, as I said, that structure then determines crystallography. Each of these elements, I've drawn two of them here, but each of these crystal units is one crystal, which is then formed of various different parts, which we call elements. And they all have, and the crystallographic orientation in this case is going along the length of the elements, and along the length of that ring. Oops. And that then gives you a radial array of calcite crystal, or a radial array, so it gives you the radial extinction cross, which probably most of you are quite familiar with. Very typical of coccolithophores, forming this sort of bright extinction cross. Okay, so that's in Emiliania, and we saw it also in coccoliths. The same process has been going on forming heterococcoliths, these ones with the complex radial subsymmetry, right back through geological time. Here we're going back down to the late Jurassic, one of my favourite parts of Britain, on Dorset, where we have this Kimmeridge Bay in the Upper Jurassic, where we have these pale, it's a thick shale sequence, actually dinoflagellate dominated, has these pale limestones, which are laminated coccolith limestones preserving blooms of coccoliths. Actually, the lamination goes down to sort of micron scale, and they're probably actually annual laminations. If you look at a broken surface, it's more or less a monospecific assemblage of what's no area. But if you look at the assemblage, well, there's a little bit of diversity in there. You can see the coccospheres, see loose coccoliths, then we can also start seeing coccoliths at intermediate growth phases. Oh, as soon as one, coming from the perspective of knowing how Amelia forms and coccoliths, you see immediately that's similar to what we saw with Amelia huxleyi. And then, if in fact, if one goes, but if you look further, you can find coccospheres like this one, which is what really confirmed it for us. That we were looking at a growth sequence where we have here a coccosphere broken open, and inside it we can see one of those coccoliths, an immature coccolith, which is still growing, or was still growing 190 million years ago before the poor thing died, and so the poor thing got it's stuck inside that. But it's a fossil record of the growth process. And so again, we can make a sequence from the coccoliths at different stages, work out the structure in a similar way to Amelia huxleyi. It's not so different at first glance to E. huxleyi. We've got growth again in various different directions to form the final radial array. But the difference here is that we have most of the coccolith is formed of one set of units, but there's also a second cycle, which you can see around here, of additional crystal units which don't join with those. In fact, if we look at it turn it upside down, here's that diagram again. This is looking at from underneath the proximal. We'll look at the proximal shield, and you actually see that around this area, we have two different crystal unit types, the ones which form the pegs. So if we extend down to the bottom, and we have the two are alternating. Well, here's again, this is a slightly dissolved specimen, here's a non-dissolved specimen, and so we have what we term the R units forming most of the coccolith, and then V units forming these little peg-like structures, which then grow up to form that. And we call them V and R because that reflects the crystallographic orientation. So the R units have radial C-axes and produce a nice bright pseudo-extinction crossing when we look at them in plan view. The V units have vertical C-axes, and so they appear dark in cross-polars. If you're not good on cross-polars and understanding what I'm meaning in those, it's really worth again reading those up. And Mario Cachau has made some really nice videos on understanding crystallography and what you see in a polarizing light microscope. Again, there's links to those from on the INA web page and other places, and I certainly recommend those to any student to have a look at those. If you have any doubts about how these images work and how you interpret what you see down the light microscope in terms of the structure which we see in SEM, then it's well worth looking at those videos. So anyway, VR. So we have these two subparts, vertical, so V units, R units, vertical and radial, and they alternate around the protococcolith ring. And that pattern is pretty cool. We first found in what's known area very rapidly. Then we started thinking this actually will be a key for us. We look at other coccolith structures, then find lots more examples. So here's another simple example from the Mesozoic Biscutum, which has Biscutum magnum, a nice big one, so it's easy to see it on it. But basically, the distal shield is formed, in this case, formed of the V units, and so dark. Proximal shield, and most of the central structure is formed of R units, and so forms a bright ring. So you see it bright in the centre, and you can just about see, if you focus up and down, you'll be able to see that the proximal shield is bright, and the two alternate. Chalcidiscus is a Cenozoic one, which forms a similar sort of pattern, with distal shield formed of V units, proximal shield formed of R units. And here this is 4 images of the same coccolith. You can see in phase contrast, the V unit is appearing dark, and it's the whole distal shield, whereas in cross-polarized light, all we can see is the proximal shield, which is substantially smaller, and it's strongly birefringent. And then, actually, for understanding a lot of the image, then this sort of sketch in side view is a quick way to summarize the structure. And again, actually in terms of making a quick mental model for understanding what the structure is and comparing them, then these simple side view drawings are really useful. And so we're showing then the V unit, which is dark in plan view, is shown dark shading, and the R unit shown bright shading. And the circle here shows where the protococcolith ring initial nucleation was. And that's one complication in Chalcidiscus, and actually in quite a few modern coccolithophores, is that the protococcolith ring ends up getting buried inside the structure. There's growth downwards from it, and so you can only see it just in here. As opposed to actually in most Mesozoic ones, then if you turn them upside down, we can see the VR, see the alternating zone where the VR unit's alternated, which is where the protococcolith ring is immediately visible on the proximal surface. I guess whenever I tap it advances. So it's there, also in VR in Privospirella, Prediscospira, Ipholithus, very different coccolith structures. but all showing the origination from nucleation here of units we alternate P and R. And so that basically ends up being a common pattern shown across heterococcolith biodiversity, and that was what got us the nice Nature paper back in '92, where basically showing that we had this common pattern of biomineralization, which of the nucleation of these alternating vertical and radial units has stayed there throughout the evolution of coccoliths, even though they've grown in different ways. And so it was a pretty cool thing, was showing how the biomineralization method in this sense is fixed, as opposed to then how it's expressed gets varied between species and between families. So that's why, in terms of biomineralization, it's an interesting process. It also, of course, then for coccolith workers, for practical taxonomy, it provides a really good key to understanding for the basis of their classification. And actually, we'd used the appearance of coccoliths in cross-polarized light and their basic structure as the basis for coccolith taxonomy way before we understood how that related to the biomineralization. But then, and that was basically when we look at there's this cool thing, not only is the biomineralization really distinctive and it defines the families, but also it's what we see in the light microscope. So it's biomineralization is often a really good tool for understanding the phylogeny of organisms, but in the case of coccoliths, we have the added bonus that this is exactly what we see when we look in cross-polarized light at them. We see the pattern we see in light reflects the biomineralization, which is the most fundamental aspect of their morphology. And so even things which can be quite morphologically different end up showing much more obvious similarities in cross-polarized light, which is why, so because our taxonomy was based on that, then actually it worked very well, the taxonomy. So it's still so now, and okay, to show that then, so when we came to do molecular genetics on coccolithophores, then unlike many groups of protists, the classical taxonomy mapped really well onto the molecular trees, because our classical taxonomy, our family level groupings, were all based primarily on coccolith structure as seen in, and particularly the structure as seen in cross-polarized light. And so, yeah, that's just showing basically the coloured. This is an early molecular tree back to 2004. It's got more complicated since there's more taxa are added, but the pattern hasn't changed. And basically, the coloured boxes here are families. And you can see that all the families basically form nice groups within the taxonomic tree. Emiliani Huxley actually ends up being a bit of an aberration now, because it's only formed of R units, as opposed to all these other ones formed of both V and R units. But if we go back to the very earliest stage, and this was right back in the '91 paper, then if you go back to the protocol ring, here's all these big units are what ends up going to form the main crystal units, and then in between them, tiny little crystal units, which actually don't grow at all. When you get to the final coccolith structure, we just basically can't see them. I've got them drawn in. There's a little grey box here, but that's almost like imagination here. We're sort of predicting they should still be there, buried in. We can't see them. Or actually, what you do see sometimes is just little slits where they would have been. So we inferred that these were V units at the time. 1991, there was no way to actually prove that. Subsequently, the actual technology for doing electron diffraction in TEM has improved, and a few years ago, then a German team, led by Ramona Hoffman, a student who did the work, applied really high-resolution TEM electron diffraction to Emiliani Huxleyi, and actually proved that way that there really was a V unit in there, which is quite nice. Actually, went and we proved the prediction. Also, on Emiliani Huxley, another thing to remember, okay, so we call them V units and R units, but it's sort of it's a shorthand that. When I say V vertical, it's more or less vertical. When I say R radial, it's more or less radial rather than being absolutely radial. And in fact, the R units particularly, they're actually more oblique than radial. You can see them here. So the C-axis is going along the hammer handle and along this direction, actually along the length of the central, the laths in the central area as well. And if you look on this image, it's nicely slightly etched, and you can begin to see that direction, this is where the R unit is, this is where the C-axis is going along this direction, and it's not radial, it's consistently rotated slightly anti-clockwise. So you've got a handedness to the structure, a chirality. Chirality actually means hand. So basically, it's a non-superimposable mirror image. You've got if you look at any coccolith, you can see aspects of any heterococcolith. You'll see aspects of their morphology which are going clockwise or anti-clockwise. So here's a Umbilicospira siebergei showing that. Sorry, Umbilicospira foliosa, and you can see that this curvature in the sutures is going this way. And if you look, every single one of these coccoliths is going the same way, and that's consistent. If you take any coccolith, then they all show the same chirality. Well, every coccolith of a given species will show the same handedness. So if you go back to Emiliani Huxley, I have tested this to destruction. I've looked at, checked it on like thousands of images of Emiliani Huxley. There's lots of different ways you can see the chirality, like this structure, and actually even going down to these little shapes of the crystal endings, and they all go the same way. And you'll never find a left. If you call that a left-handed form, you'll never find a right-handed form of Emiliani Huxley. You'll never find an Umbilicospira foliosa where the curvature is going anti-clockwise instead of clockwise. If you do, I will give you a prize or something. But it's a confident prediction you won't. We've looked at lots and lots of specimens without ever finding it. That also gives a clue to where that chirality comes from. In some organisms, like snails, you have. You remember that gastropods, the vast majority of gastropods will curve one way, but you get an occasional one which goes the other way. Same with foraminifera. You get that foraminifera, typically a foramen species will coil one way, but then maybe 1% will coil the opposite direction, or maybe in some species even 50-50. And that's because the morphology there is developing by cells adding on. And so basically, by chambers being added, and it's a sort of supramolecular process, a fairly large-scale process. In the coccoliths, then the completely consistent chirality suggests it's actually being controlled at the molecular level, because when you go to the molecular level, then basically all molecules are chiral. All the large molecules are. So DNA is chiral. Every single DNA molecule spirals the same way. Otherwise, this wouldn't work. And so then the proteins which are coded by DNA, when proteins are formed, again, it's a chiral structure. Every single protein goes the same way. And so in consequence, when you go down to the real cellular, when you go down to the bottom of biochemistry, then everything is chiral. And then, so if you have consistent chirality, it's an indication it's basically being controlled at the molecular level. And so that then leads to prediction that probably what's controlling the nucleation is some kind of molecular template. So in some way, so basic idea, we have some kind of macromolecule which has binding sites which mirror the binding sites on a calcite. Click on a calcite in calcite. So you've got mirror the ionic array in calcite. And so actually, so Mann and Sparks 88 suggesting there's some kind of template which accounted for the fact that all the Emiliani Huxley crystals were perfectly arranged, perfectly aligned, completely controlled in terms of both their C-axis, which is the optic axis, but also the A-axis. So it's completely aligned crystals. Extended that a bit further, we suggested back in the 92 paper that maybe there was some kind of folded template which then could produce alternating vertical and radial rays. And Mary Marsh subsequently made a sort of slightly simpler but less, in some ways less elegant way of folding them in order to get the same kind of effect. Still think that's probably true. In this case, we've had the predictions been lying around for 30 odd years now. And we haven't actually managed to identify and find the protein yet and find out how that templating goes. But we still seems reasonable to think there's some kind of molecular template. Okay, on to some complications of the model. First one, central areas. The simplest pattern is for the so okay. We have the rim and the central area. Essentially, the rim is what's formed by growing outwards from the protococcolith ring, and the central area is what's formed by growing inwards. And in some cases, its central area is just formed of the same elements. So in Emiliani Huxleyi and other reticulofenestrids, the grill in the centre is just formed of the same units growing radially inwards, and even the bridge here in Japhira capsa is just formed by the same units. There's no nucleation, just some of the ones of the inner tube just arch over and form the bridge. And so you can see how you can actually get quite different morphologies just by allowing the growth. You've got the same, exactly the same basic biomineralization pattern, but just allowing it to grow in different ways, you can make what sort of species and generic level sort of differences. So again, of course, in some of them we get slits as well. That's a simple one. Other ones, though, start being a bit different. It's that they don't form entirely by the simple. So in coccoliths, we've got, this is an early growth phase of coccoliths, just to show the structure. This will end up turning into big coccoliths by the shingles growing outwards. But across the central area, we have the bridge forming here. And you can see here that the bridge is formed of separate little rhombohedral crystals. So in this case, the central structure is formed by additional nucleation. But it's sort of like VR nucleation. Quite often when you look at those sorts of central structures, and quite a lot of Mesozoic ones show this, you actually see alternating two types of crystals alternating go along that central structure. So that's sort of essentially I see this as being, it's VR nucleation, but we've got one belt of VR nucleation around the rim and a second one going across the center. Then you can have something significantly different, which is what happens in, say, Rhabdospiraceae, where they basically got VR mode in the rim, but in the central area, they're just doing something totally different. And we don't actually, this is an aspect, again, which we just don't understand. We don't know quite what's happening here. But it can be amazingly cool. There are some quite phenomenal structures. And they also, I mean, not only are they phenomenally complicated and beautiful, but also just like each one seems to be different. Each genus, they go and reinvent how to do the central area structure. So here's Rhabdospira, this quite awesome structure. We've basically got 5 spiraling sets of rhombohedra, and each one of these is a separately nucleated calcite crystal, but they end up with this 5-pointed star and a little pimple on the end. Then Discospira, again, really beautiful structure. Yeah, there's a really nice image on the NannoTalks poster, which hopefully you've seen, showing it, how we've got this very cool sort of funnel structure, but that's being formed by a completely different process. It's just the outer part of the rim which is formed with the VR, the rest is a different structure, and it's still forming inside the cell though, which is what you can see nicely on this image, where this trumpet here is still inside the coccosphere. And so the trumpet and the base are both formed inside the coccosphere, and it sort of almost looks in this case like it's too big to go inside the coccosphere. And part of the reason is actually in Discospira, they seem to form the trumpet and the base separately, and they're only joined together once it's exocytosed. And no, we don't fully understand how that happens. When we get through into the complication, there's still lots we don't understand. Okay. Second complication on VR is that model only applies to heterococcoliths, the ones with rims with complicated crystals. There's a whole separate group of coccoliths called holococcoliths, which are formed just of a mass of minute little rhombohedral crystals. Each individual crystal here each of these little rhombohedra is a separate crystal. The crystals have very simple morphology. They're just rhombohedra, like just simple calcite rhombohedra, just like you get if you growing calcite inorganically. But they're all the same size, and then they're beautifully arranged in tiers and structures and nice sheet-like arrays to make the final coccolith morphology. And it's a very different biomineralization process. And quite logically, when people first realized we got these two different types of coccoliths, holococcoliths, heterococcoliths, it was assumed they were just different families of coccoliths, taxonomically different groups. Then in 1960, Park and Adams, back in Plymouth, biologists in Plymouth had a culture of Coccoliths pelagicus, this form Coccoliths pelagicus, and then it suddenly started changing to forming this sort of cell with much more delicate coccoliths, as you can see in light microscope. It's forming a very thin layer around the outside. Then when they looked at that in the transmission electron microscope, they found it was producing a completely different type of, it was actually producing holococcoliths. And they were able to do this repeatedly, so they absolutely knew this. It wasn't any contamination. It's the same organism, basically can undergo a change in its life cycle and produce a different type of cell. In this case, it also swims. You can see here the flagellae have a motile phase, the life cycle, which produces holococcoliths, as opposed to a non-motile form, which forms heterococcoliths. And then actually, if you go out to the Arctic particularly, you'll reasonably often, find coccospheres like this, where we've got the heterococcolith forming phase which is covered also with holococcoliths. So what's happened is you've had a life cycle change, and it's gone from the phase which produces holococcoliths to the phase producing heterococcoliths. We call these combination coccospheres, and occasionally you find them in Coccoliths pelagicus examples are reasonably common. There's a few other which come on. Other ones they occur much more sparsely, but by observing these, which was started by Louisa Cross back around 2000, really started highlighting the value of these for working out life cycle associations. And here's a lovely example of hers from Helicospira carteri, the heterococcolith phase, which forms these amazing spiral structures. And again, every single one of these always goes the same way. And then you occasionally find these combination coccospheres, which show that the alternate life cycle phase is this type of holococcolith. And the cartoon here is sort of showing the idea that you have two holococcoliths which have a haploid cell, but only only one set of chromosomes will then fuse to produce a new compound cell, and that new cell will then start producing the heterococcoliths, and eventually and then end up as this sort of cell. And that's a nice example where you've got multiple images. But then basically we start finding occasional examples in lots of other species. Here's Siracospira, and yeah, there was a phase of these back to the early 2000s, but they're still periodically we find another example of it, and it's slowly been building up our knowledge of how the two tie together. But we also that thing of how it works, the life cycle. There's a few of them where we've got them in culture, and we've got both phases in culture. And when they've got that, they will very occasionally change, but most of the time you just have a culture which forms only one type of cell. And then what you can do is measure the amount of DNA inside the cells. And what we find is there's double the DNA in the heterococcolith phases, have double the DNA of the holococcolith phases. So this is the haploid phase with one set of chromosomes, as opposed to the diploid phase with two sets of chromosomes. That's the summary diagram. Diploid phase producing the heterococcoliths, and it will carry on growing asexually happily for in the case of something like the Calcite discus, years and years in culture, and then until for some reason it changes and it then undergoes meiosis, produces the haploid cells, which again will then produce the holococcoliths, and again they will reproduce asexually for ages. So you've got two separate stages of the life cycle, and then just occasionally we find the transitional coccosphere. So in terms of ecology and things, it's a really important aspect. It's also, in terms of the biomineralization, it's something quite extraordinary, this, where for some reason we've got calcification occurs in both phases of the life cycles, but with different mechanisms. And those two mechanisms have stayed stable through the evolutionary history of the coccolithophores. And that's something we really don't understand. It actually makes a really interesting system in terms of genomics as well, because you start trying to see what genes are expressed in the different phases and use it as another way of trying to understand how the biomineralization works. And okay, the holococcoliths, we really don't understand the biomineralization. And one thing that holococcoliths on is the biomineralization is much more flexible and anarchic than it is in the holococcoliths. So at species level, the morphologies are really defined, and you sort of can define genera of holococcoliths, but they don't tell us the large-scale classification, anything like, unlike heterococcoliths, where we can predict phylogenetic relationships from, if they've got similar coccolith ultrastructure and VR pattern, and we can predict they're related. In the holococcoliths, it just doesn't work. Okay. Final complication, which I thought I should throw in this version of the talk, is nanoliths. So we've got holococcoliths, we've got heterococcoliths, both of which are produced by Coccoliths pelagicus, both of which are coccoliths, which we sort of define coccoliths as being things which are produced in a homologous way to the coccoliths formed by Coccoliths pelagicus, since that's the original one. But then nanoliths are ones which show the same sort of they're again calcareous nanofossils, the same size as coccoliths, and showing complex regular structures, but right down to crystal level. But they don't show either V mode or R mode either VR mode calcification or holococcolith calcification. So they don't fit either pattern. And so then there's three possibilities basically for nanoliths. One possibility is that they're just heterococcoliths which have got different and lost their, so the VR structure is no longer obvious in them. The other one is there might be holococcoliths, where the holococcolith structure is no longer obvious, or there may be something completely different. And almost certainly within the Nanoliths, there's examples of all three. Florispira profunda is something we call a nanolith because it's basically just a single big calcite crystal. The whole coccolith is basically just one crystal unit. But in fact, if you look at the bottom of them, if they're really nicely preserved, we find they've got tiny little crystals. So basically what's happened is it's a not regular heterococcolith, which is what decided it wants to turn into a completely different shape, and it's evolved that by basically just taking one unit of the rim. It's where it's the axis going that way. So it's a V unit, although it falls over, so it's having birefringent, and it's basically just, so they've taken one element and it's grown up into a completely different shape. So it's another example about how typical sort of way evolution works. If you're going to design a coccolith from scratch, you're going to design a Florispira nanolith from scratch, you'd never bother with the holococcolith on the bottom, because it's evolved from a heterococcolith, it still has it attached to the bottom. So this case, profunda is most definitely, it's a modified heterococcolith. And there's quite a few other examples back in the fossil record, particularly where we know that's the case. So for instance, Lithraphidites is long rod-shaped thing, but we find tiny little basal coccoliths on them. And there's quite a few cases where nanoliths essentially are just modified heterococcoliths. Other cases, they definitely aren't. So Ceratolithus, we know from the life cycle, sorry about the slightly garish colours on this one, but Ceratolithus has two phases in the life cycle. One phase produces heterococcoliths, the other phase produces the ceratoliths, which definitely aren't heterococcoliths, and it's a reasonable prediction they probably are produced in a haploid phase. Well, there's a bit of a complication. They also have these hoop coccoliths which are produced in the. So we're a bit unsure about exactly how the haploid diploid phase works, but they're not just modified heterococcoliths, because these are the heterococcoliths we can see on the outside. That's heterococcoliths. These form the nanoliths, whatever they are, they're not modified heterococcoliths. And really, it's hard to see them as being in any way a heterococcolith. And also, when you have a cell, the actual cell is smaller. So in some ways, it looks like they're being formed outside the coccolith. Another example where we know it's it's actually neither modified holococcolith. I mean, it's conceivable in some ways that you could describe as a modified. It might have evolved in some way from a holococcolith phase. Polycrater is another one. Polycrater, you'll only come across these if you work on modern plankton. They're really small. It's only about 0.2 of a micron across, little square cups. And these are made of aragonite, not calcite, which is one clue that they're not homologous. And then the alternate life cycle phase, we know again from the work of Louisa Crosses, they form their alternate life cycle phase is Alispira, which is a completely normal heterococcolith. No, I haven't got a picture of it. But basically, it's a perfectly regular heterococcolith, and the alternate life cycle phase is an aragonitic thing which looks sort of like a heterococcolith, but it doesn't look like a holococcolith. So again, it just seems like this is it's not a modified holococcolith in any way. They're weird things. So it looks like in this case we've got another totally separate biomineralization mode. So then it's totally reasonable to still call them nanoliths as being not holococcoliths, not heterococcoliths. They're something else, even though they're being formed by the same taxonomy. They're still being formed by coccolithophores. And another example on the same lines, Verudospira, everybody, one of our favourite species, Verudospira bigelowii, produces these beautiful pentagonal nanoliths, which then form a nice dodecahedral sphere. And we know from beautiful work, particularly by Kyoko Hagino, that they're definitely not heterococcoliths. On the one hand, so we know the alternate life cycle phase in this case is a haptophyte which non-calcifies. The calcification occurs around the outside of the coccolith, they aren't formed inside the cell and exocytose. In terms of the biomineralization, they're very different. They've got tangential calcite C-axes rather than radial, and then these 5 units each behave as a single crystal unit with a tangential C-axis. But then if we look at them in side view, they've got this laminar substructure to them. They're formed fine little laminae, and they sort of seem to grow by stacking up laminae. So they're growing in a very different way to either heterococcoliths or holococcoliths. So again, that's a different biomineralization mode, which seems to have evolved separately. So it sort of begins to look like, in some way, within the haptophytes, we've got a group. Oh, okay. And then the molecular genetics. Basically, here's all the holococcoliths. Here's all the coccolithophore clade, and then the Brudospira on this is plotting really basal in the tree. In this tree, it looks like they're forming inside the coccolithophore clade. In other ones, they fall just outside it. But very broadly, the pattern we see is we've got calcification occurs distinctively in the haptophytes, and it's always within actually one sub-part of the haptophytes, which is what we've now, what name we've given it for this is the calcihaptophytes. The haptophytes which have the ability to calcify. Some way they evolved the ability to calcify, but then it's been expressed in different ways, both to a certain extent within different groups like Brudospira versus the rest of the coccolithophores, and then also expressed differently in different life cycles. So we have it expressed differently in the haploid and the diploid phase. So that just about thank you for listening. And to summarize, coccolith calcification is a very, very controlled process. It's a really fundamental aspect of but calcification is not easy to do, both because you've got to modify the cell biology quite a lot in order to control, to actually get the components into the cell and then to regulate it, but also because calcite itself is a very anisotropic substance. So to shape calcite into a coccolith takes really a lot of biological control. It's a very controlled process, and in consequence of that, patterns of biomineralization make very good taxonomic characters. And so it's really useful it's a fundamental basis to understanding the taxonomy and recognizing how we classify coccoliths. So yeah, crystal orientation are really useful characters. And just to recap, the main biomineralization modes are heterococcoliths, with start off with radial arrays of VNR units, holococcoliths with minute rhombohedral crystallites, and then we have nanoliths, which have variable origins. So, hope you found that interesting. And as I mentioned in the talk, if you want to understand more about the crystallography and how in practice you start look at the morphology, I do recommend Mario Cachao's Crystals for Nanos movies. Thank you.